about 30% of all of the carbon dioxide that we've added to the atmosphere so far. So on the one hand, this is great. All that carbon dioxide that's in the ocean isn't in the atmosphere, and that's slowing down the rate of climate change that we're seeing so far. However, it turns out there are also some negative sides to the ocean absorbing carbon dioxide from the atmosphere, because as carbon dioxide is absorbed into the water, it forms a weak acid, and that's changing the chemistry of the oceans throughout the world. And some of you might have seen a recent series in the Seattle Times talking about this problem, which is called ocean acidification. And this is something we care a lot about in the Pacific Northwest, because these changes in ocean chemistry have the potential to be really harmful for organisms we care a lot about around here, such as the Alaskan crab and oysters that are farmed on the Washington and Oregon coasts. So this is something with really important potential impacts on the local economy. So what I'm trying to understand is the mechanism by which the ocean is able to absorb this carbon dioxide from the atmosphere. And hopefully that'll help us better predict how much more carbon dioxide the ocean will absorb in the future, so that we can better predict both what's going to happen with global climate and what's going to happen with these changes in the chemistry of the ocean. And I'm particularly interested in how much of this absorption of carbon dioxide is done by physical and chemical mechanisms, and how much of it is done by biology. And I get really excited about the biology because it's something that we don't understand very well so far. Some of you, if you've flown on a plane and thought, well, maybe I should buy some carbon offsets so that my emissions aren't going to affect the world, you might have bought some trees to be planted in a rainforest. And so people are pretty aware of the fact that photosynthesis by plants on land take up carbon dioxide from the atmosphere and store that as organic carbon. But most people don't realize that half of all the photosynthesis that happens on Earth actually is happening in the ocean, is being done by these really microscopic organisms called phytoplankton. So I'm really interested in the potential role of these phytoplankton in taking up carbon dioxide from the atmosphere and then storing that carbon deeper in the ocean, where it's going to both change ocean chemistry, but take that carbon dioxide away from the atmosphere and slow down the rate of climate change. And so to do this as an oceanographer, I have to go out to the ocean and collect samples. And the way I do this is pretty unusual, even for an oceanographer, because I ride on Chinese container ships. So I fly from Seattle to Hong Kong, and I get on a Chinese container ship, which is about the length of 3 football fields. And I'm the only scientist on the ship, usually the only woman. And I ride the ship all the way from Hong Kong to Long Beach, California. And for 2 weeks while I'm on that crossing, I'm continuously making measurements of how much carbon dioxide the ocean is absorbing, the role that's being played by photosynthesis in the surface ocean, and who are those phytoplankton out there that are doing photosynthesis, so that we can help better understand the role that they're playing in global climate. So I'm really excited about the work that I'm doing right now, but you wouldn't necessarily guess that I grew up landlocked in Pittsburgh, Pennsylvania, with no real contact with the ocean until I was in college. So it wasn't until the end of my undergraduate studies that I realized that this was what I wanted to do with my life. And so when I applied to graduate schools, I really looked all over the country, and I was convinced that the University of Washington was the place that I wanted to go. But it was a pretty big decision to decide to move all the way to the West Coast for my graduate studies. And so when I found out that my department was offering me an ARCS fellowship, they were putting that support behind me. I would have some extra income to make the move across the country a little bit easier, and then found out that I had this whole group of supporters who were out there who were going to cheer me on throughout my research, and then got to meet Ann Zubko and her husband Michael, who have been amazing supporters throughout my time here. It's really made it a lot easier for me to transition into the field of oceanography and be able to be successful in my research. And so I'm really excited about the work that I'm doing, and I'm also really excited about being able to combine research with teaching, because what I've learned over my years studying climate as a graduate student is that this is not a problem that is going to be solved just in my generation. This is a problem that's going to even require the next generation of scientists after me. So I really appreciate the support that you've given me so far, and I hope that I'll be able to then help inspire and train the next generation of scientists to solve this problem. Thank you.
Thank you, Hillary. My name is Cole Muth, and I'm from St. Louis, Missouri. I decided to do a PhD in plant science because I think it's really one of the key research issues of the 21st century. People in developing countries are experiencing an increase in their income, and they're wanting both more food and a greater variety of food in their diets. And at the same time, there are other areas of the world that have terrible food insecurity. So how do you provide for this added demand on a quantity of agricultural land that's essentially fixed? Well, I think that a great place to start is the crop diseases that threaten farmers' livelihoods all around the world. So I am in the plant pathology department at WSU. I work for Dr. Scott Hulbert, who's been gracious enough to be here in the audience with us today. Hi, Scott. And we study wheat rust fungus, which is a very serious parasite of wheat. And if you know any farmers in the Pacific Northwest, you may have heard them talking about stripe rust and how expensive it can be to treat with fungicides. So it's an expensive problem here in the United States, but it is absolutely life-threatening for folks in poorer countries who don't have access to the kinds of technologies and fungicides that we have here. And untreated, wheat rust can take anywhere between 10 and 70% of the entire wheat harvest for a country. And in fact, that's exactly what happened in 2010 in countries like Syria, Ethiopia, Uzbekistan, Afghanistan. These are countries that definitely do not need added food insecurity, because there can be no political progress when the price of bread is astronomical because the wheat harvest was especially poor. So in dealing with this problem, I think the solution lies in the answer to a really fundamental question, which is, why are certain varieties of wheat naturally quite resistant to stripe rust and others are very susceptible? And it's not just so simple to say, well, let's plant the resistant ones and avoid the susceptible ones, because the fungus can very quickly evolve resistance around this natural genetic resistance that the plant has. And so our job is to kind of understand this two-way interaction between resistance genes in the host and virulence genes in the pathogen. And we hope that once we have that figured out, we can start to design varieties of wheat with durable resistance to stripe rust that will optimize yield on a given land area and that can minimize the use of pesticides. I think I have a really great job as a grad student because I get to think about these fundamental questions in plant genetics and biochemistry, and then when we discover something interesting, the application is often quite clear. And that's the kind of translational research that drew me to Washington State in the first place. I admit I had a really hard time deciding on where I would ultimately go to grad school. I applied to several places, and I managed to get into Purdue in Indiana, and University of Illinois in Urbana-Champaign, and also Washington State University. These are all fantastic schools for agriculture, as any principal investigator will tell you. But one difference among them is that, honestly, Purdue and Illinois offer their grad students a lot more in terms of the stipend. And so I didn't want my decision as to where to go to grad school to be based on that at all. And so I called up the program director of molecular plant sciences, and I said, do you have any scholarships or fellowships that are available? And he said, yes, absolutely. There's this ARCS Foundation fellowship, and you're perfect for it. And this will allow you to make your decision just based on the science that you've observed and the people that you've met here during recruitment. And so that's what I did, and here I am, and I really want to take this opportunity to express my gratitude to the Seattle chapter of the ARCS Foundation for their support. Thanks.
Aren't we lucky to hear from such amazing students? Thank you. I think I think we'll all look forward to hearing about their careers in the future. Well, at this point, I would like to introduce our keynote speaker. It's very exciting to me. His name is Nathan Kutz. He's a professor in the Applied Mathematics Department at the University of Washington, which happens to be ranked #1 in the country. Professor Kutz received his BS in Physics and Mathematics at the University of Washington in 1990. He received his PhD in Applied Mathematics from Northwestern University in 1994. He joined the University of Washington's Applied Mathematics Department in 1998 and became chair in 2007. Professor Kutz is especially interested in a unified approach to applied mathematics that includes modeling, computation, and analysis. He has held many impressive academic positions and has received numerous awards and honors, so many so we would be here all day if I were to mention them all. He's just returned this summer from MIT and Harvard University, where he was a visiting professor. Please welcome our guest speaker, Nathan Kutz.
Well, my mom would love to hear that list, I'm sure. So what I thought I'd do is tell you a little bit about what applied math is, first of all, because I think a lot of people ask, okay, they kind of know what math is, but there's also applied math, there's statistics, computer science, and all of us, by the way, these 4 departments are exceptional here at the University of Washington. And they've really had a huge impact on technology-driven companies around the area. So I just want to put that out there in terms of when you think about the mathematical sciences just up the street and how it's affecting the state of Washington, it's really enormous. You can never have too many people with good math skills in your state. Let's start off there. That's important to recognize. So, okay, we'll go to my slides. That's a way too big a picture of me. All right. So you can't quite read, whenever you save different PDFs, it mess up. But I was talking here about this idea of perception, decision-making, and all the way to apps. And part of what I want to do is illustrate a little bit of what applied math does, but also make connection to some of my colleagues in this room. So let me flip forward a slide here. Okay, so first let's talk about applied math. We just moved this March into a new building, Lewis Hall, and there it is. It used to be a men's dorm, turn of the century. And in fact, what I really like about what has happened with this building, it's the 3rd oldest building on this campus up the road here. And I feel like what they have now is the caretaker they've been looking for. This has been, various groups have been rotating through this 5-10-year cycles, and now we're there, and we're going to make this a great home for applied math and grow it into a great program. We already have a great program. There's our little brag, the National Research Council rankings. We were tied for first, but we can just ignore the Princeton there. And so this is quite an achievement, especially as you look at rankings and you look at sort of the funding level and the ability that some of these privates have in terms of the cash flow. And for us as a state school, if you look around, I'm going to come back to this point. It's all about people. It's a simple message, really, and it's all about people. It's about people at the faculty level. It's about bringing the best grad students in. And that's what we can do, and that's what we use this for. We have a lot of different skill sets in applied mathematics. We do a lot of scientific computing, high-performance computing, as we know in this area in Seattle. People like computers. People do a lot with computers. There's a lot of industry around that, and we solve science problems with them. We also do a lot of mathematical biology. This includes studies of ecology, a lot of neuroscience, and just general things like what are generally called nonlinear waves. We have some people working on modeling of tsunamis. In fact, I think you've maybe saw in the paper the other day one of the first schools that has now a tsunami plan in their gym about going up. And this was some studies done by mathematical computations of what would happen under if a coastline were inundated. So these are the kind of things that applied math can bring to the table. We engage deeply in scientific areas, and but we also try to do very careful, bringing it all the way back to very strong mathematical foundations. And I want to talk a little bit about what I have a great deal of interest in. Neuromechanics, or reduction of complex systems and neurosensory systems. Now, I want to be, in fairness of disclosure, I want to tell you how I got onto this problem. As a few years ago, in that lobby, I was feeling a bit antisocial. And you guys were all there, and I know there was a bunch of people probably feeling this. Okay, all these people, I gotta mix and mingle. And that particular day, I didn't feel like mixing and mingle very much. So I already done a couple fake bathroom runs. That got me 5 minutes, and I'm not even gonna ask for a show of hands how many people did that already today. But you do that, and you're kind of running out of steam. I can't do that one again. And then I say, okay, I'll go get a fake coffee run. I just had helped this cop. I'd done it once. I'll go back over there. And as I went to the coffee table, which now is located over there, but it's right outside that door before, I happened to run into a group, and I was forced to socialize. But one of the people that I ended up socializing with that day is sitting right over at table 19, Jeff Riffle, who was a new faculty member in biology. And he started telling me a little bit about moths and their olfaction processing. We started getting this conversation, and Nathan was no longer feeling quite so antisocial. And Nathan was inviting Jeff, come to my office, bring your slides. We'll sit down, I'll make you coffee. That actually started, what do you call it, the virtuous circle? That's the buzzword these days. We started our own little virtual circle right there in that lobby. So good things can happen at ARCS in addition to funding of students here. So this is how I got into this. Now, some of you may or may not know about neuroscience, and what I thought I'd do is at least convey to you the idea that you actually have a very good intuitive feel for some of the things we are trying to address. And by the way, some of the things that we are trying to address, which is this idea of decision making. How do we take information input, process it, and make decisions based upon it, right? And we have floods of information, yet sometimes these decisions are made on very simple things, okay? And we were not the first to address this. Not the computer scientists, not the biologists. We start off, I think, with the artists. I give you Picasso and his dog. So there you go. By the way, everybody sees that's a dog? This is an exact example of what I'm talking about. I give you almost no detail of that dog. And it didn't take you more than a split second, and you knew, oh, there's a dog. Not only did you know that it was a dog, you knew out of all the brands, or whatever they're called. I'm a mathematician, not a biologist. My kids are really pushing hard for a dog, and I have to give in at some point, but not yet. But that is your dachshund, right? Now, I found out more about these pictures. I don't know. Picasso actually did a bunch of sketches like this. And you know why he did this? This is fantastic. He was Picasso. He was famous. And so he'd go to these restaurants in Spain, and Spain has fantastic food, and I'm sure he only went to the best places. But, the bill comes at the end of the night, and he didn't want to pay it, so he would sketch these things on a napkin, and there's your payment. So, with that said, we're going to hand out Sharpies and take the best picture and see if we can give it to the Sheridan and see if they cover this lunch. So start thinking about what you want to draw. Somehow this really represents so much of what we want to get after, is how do we do this? How do we process that? And do we really need details? Or is it enough to have sort of this core pieces of information? Let me give you some more example. I'm going to bury something in an image, and you're going to try to extract it. This was at the SAM not too long ago. I give it to you. I'm going to give you a few seconds to look at it, and then I'm going to give you the answer. Okay. All right. So this is kind of a little like a roller shade test, but here we go. The guitar player. And I don't know if you can see it. 2 elbows out. You can see the fretboard. Here, I got a laser pointer. Let me put my laser pointer on it. Here. See the fretboard right there? Elbow, and this is kind of the head region. So Picasso played a lot with this idea of being able to pull in information and kind of make something meaningful out of what looks like just a bunch of lines and colors put together. Here's another one, more popular culture. It's George Washington, not Jiroji. But here, this is what I love about this. This thing here was on eBay, and it actually sold for over $8,000. Okay? And it was 3 years old, by the way. Someone got this George Washington chicken McNugget and then sold it. By the way, so this was a Seattle Times article. Oh, let's go back one. But, here's another one. Right around the time of the wedding. The Kate Middleton jelly bean for 1,000 pounds. I'll sell it to you for 2. Anybody? Takers? So for the cost of, ARCS fellowships, you could have a jelly bean or a McNugget, or you could help someone develop science in the future. And that's, by the way, from the CNN webpage. By the way, I don't know if you look at anything as trashy as CNN.com, which I do. Every 2 months, without fail, someone saw, Mary in a pancake or this or that. There is something out there in a sandwich, on a burrito, in a cloud, without fail. 2 months. They're just like, okay, who saw something? We got to get that article out there. So, but part of this is interesting, right? Because it tells us something important about what we're doing. We're born to recognize things. We have a lot of things. We're trying to process this information. It's not like I will decide to look at this and try to see if I see something. We just see it. And this is something that we have built into us. So we started exploring this in this moth called the Menduca sexta. Now, there's a little moth farm up the road that Jeff has, and there's Jeff's nice picture of it. And it's really interesting when you can take this moth and close your ears if you're faint of heart. You take its head off, and you put it on a petri dish, I think, or some substrate, and you can then take it open and put recordings in there and start recording this thing as you give it odor stimulus. Okay, and this thing will stay alive for a day or two, okay, if you take very good care of it, because there's no blood circulation. So you have this moth head, and you're taking recordings, okay? And what's interesting is you apply odors here, different odors. If you give it some odors it likes, you see some of the neurons really go crazy. It's somehow encoding really important information about that odor. And then so you take all these recordings, take all this data, and you start to try to understand how it's processing this data. And then afterwards, you can throw it in the trash. Clean greens. I think we can clean green this, right, Jeff? Can you clean that? Yeah, yard waste. So that's it. And then you go through the next one, and you start taking all this data, and you start building a story about what the processing unit of this moth is doing, because it's trying to do something very similar to what we're doing, which is take this information in, recognize, is it a flower that I like? Is it something else that I like? Is it a female pheromone? It reacts to all these things. And this is its main processing unit to drive its behavior. So here's a slide. It's a little bit, maybe too much. But I'm just going to give you an example of real data. So we're trying to connect, again, real biological data to a real application to using all of our most sophisticated mathematical skill set, right? This is what I think of as the applied math story and also interdisciplinary story at the University of Washington. And what you see here is that if I give it some odor A at a neuron over here, and I put a probe right there, and I give it this odor, what happens is the neuron's firing away, and as soon as I apply that odor, the neuron shuts down. It just goes quiet. And about a second later, it comes back on. But if I apply odor B to that same neuron, look what happens. It's going along. As soon as I apply the odor, I get this burst of activity. It's almost like that neuron clearly recognizes that chemical composition of that odor. The opposite happens over here at this neuron, which is nothing's going on. As soon as I apply odor A, it goes crazy. And I apply odor B, shuts it off. So these opposite behaviors for odor A and B in these neurons. And what you could do is take lots of recording around this antennal lobe, and what you find are these patterns of activity that are robustly produced for different stimulus. And these somehow are the keys to understanding this neurosensory processing that's going on. If you will, this pattern somehow here is somehow, that's like your recognition of that dog. You don't have to have precision about building that odor. You have to just know that it is, in fact, a dog or a cat, or these aren't smelling dogs and cats, they're smelling certain types of flowers and female pheromone. And so you see right away there's this very interesting architecture that happens here of data processing, and it's dynamical, and you want to try to understand it. Well, this is how you get at it. You need math, you need neuro, you need leading edge skill sets in both. And it's a great, great fun project that we've been working on. So the other thing that's kind of great you could do with these is you can understand how learning happens. So Jeff can take this moth head, and its proboscis is still there, right? So this is this head with a proboscis, and you can reward it, reward the head. So you can give it something like a new odor and sugar water at the same time. And within 5 to 10 tries, this thing learns a new code for whatever odor you put in there. So I've been trying to push this on Jeff. He hasn't bought into it yet. I give you a cappuccino. This isn't my office, by the way. I'm a little bit of a cappuccino snob, so that's my own artwork right there, fern leaf. So what I have this idea of, you could take this moth on your shoulder, be walking around town, have it trained only for the high-end coffee, send it off, have it go out, and you've trained it, and it's going to go find you the high-end coffee, and all you got to do is follow this moth right to the best coffee shop in town. And then that's how you go to a new city. And you don't know how to take recommendations, you just take your moth with you. I think we could sell these $99.99. eBay it, just give us your address. But this is very interesting. It allows us to study very fundamental things that moths do, like this learning. It also is starting to lead us to think about the opposite of learning, which is something like concussion or traumatic brain injuries, which are very important to understand, in which you damage this network of activity. And so that's one of the things we're also thinking about, is how does this structure, how is it able to process information when you come into this antennal lobe region and you damage the connections? And this is very important. People really would like to understand this, and obviously you can't do this in humans, but here's a test model where you can maybe start understanding how functionality works or is compromised under these kind of things. Now, it's not just biology that you could apply these ideas to. After all, what we did is we did this data processing to try and understand key features of what's going on in this. I'm going to give you another example here of an ARCS fellow that was working with me, Jake Grosick, and we were trying to think about, okay, and this is a long-standing problem. We were just looking at gesture recognition and thinking about how do you turn your camera on your phone or your laptop, and instead of having your mouse, you could just control it with your hand. You know, you could go to the next, scroll to the next page, do a one-click, a two-click, stop. You could do everything with your hand, just turning on the camera. And so these are kind of things that people would really like to understand how to do. So for instance, even something like this, where you want to just train it to do, I want to basically do away with this mouse and just turn that camera on right there and be able to do these different gestures and replace my mouse with my hand gestures. Okay? So I could give this talk, and if I want to go to the next slide, I just do this. Things like this. This will happen. It will come. But it's still out of reach at this point. But how do we understand how to do this? Well, let me show you one example of this. And I can ask you, what is this? I'm going to tell you, this is one of those gestures. And in particular, I took these gestures and at 8x8 resolution, this is this. You can hardly tell what this is, but I tell you what, my computer knows what that is with the accuracy of 99%. This is the kind of things you can start thinking about with computer vision. How do I take all this data? I can compress it down to simply 8x8, make a very quick classification. So you have this idea of being able to do very fast computation. And it works a lot like the dog problem, right? So you start seeing that there's this commonality, whether it's neuroscience or whether it's computer vision type applications. What you're trying to do is make recognition decisions. And this is where you can start thinking about developing app-type structures to think about incorporating these things into devices such as this. I don't know if you love your iPhone as much as me, but I love my iPhone. I love the apps. I love all the stuff you can do with it. Sometimes you just need to put it away. My wife doesn't like my iPhone that much. I'm sure some of your wives and husbands don't like your iPhone that much either. But these are the things that you can do. There's amazing still potential for functionality. So how do we make this all work? And I made this comment and earlier, it's all about people. What the University of Washington is able to do is in our departments, so many of our departments are so good, same thing with WSU, what we have, everything is going to drive these universities forward are going to be the people we bring there. I don't care if you have the nicest campus,